chloroplasts, that's the green pigment. Schimper in 1883 coined the term chloroplastid for green plastids. Mayer called them as autoplast. Chloroplast name was proposed by Herrera. Grana, made up of grana lamellae arranged in the form of stacks of coins. Each grana lamellae is surrounded by a well-defined membrane called as thylakoid, and you know that each chloroplast has 40 to 60 grana. Quantasome, you know these are the distinct photosynthetic units. It was first observed by Park, Pon and Beggins, and each quantasome consists of 230 chlorophyll molecules. Each chlorophyll molecule has two parts. The head, which is of course is a tetrapyrrole ring, and a phytol tail. The head part is responsible for the green color, which has a magnesium atom in the center. You can see the chloroplast, the grana, you can see the thylakoids, outer and inner membrane, stroma lamellae. Due to the presence of chlorophyll in granum lamellae, the light reaction of photosynthesis is connected with the granum, and lamellae which connects grana are called stroma thylakoids or Fred channels by Weir, 1966. Stroma, the non-pigmented enzymatic part of chloroplast, is called stroma, and it is present in form of matrix. And due to presence of enzyme in stroma, dark reaction of photosynthesis takes place. That is, you can say there is a division of labor. That granum is concerned with the light reaction, and the stroma is connected with the dark reaction. Stroma of chloroplast contains proteins, phospholipids, nucleic acid, ribosome, and less amount of enzymes. Riss and Plot, Ruth Sagar, discovered DNA in the chloroplast, which is of course called as ctDNA or plastidial DNA. The genetic system of chloroplast is called as plastidome. Due to the presence of DNA, RNA, and ribosomes, chloroplast, like mitochondria, is called semi-autonomous unit, like mitochondria, having a complete protein synthesis apparatus in the stroma. Now comes the ribosome. Ribosomes are the smallest cell organelles, and the size is 150 to 250 angstrom, unit membrane-less organelle. Pelletier coined the term ribosome. In plant, Robinson and Brown were the first to observe them in the bean roots. Except mammalian RBC, all living cells have ribosome. Ribosomes are universal cell organelle present both in prokaryotes as well as in eukaryotes. Here you can see the ribosome. Types of ribosomes. Prokaryotic ribosomes, their 70S, occur in the cytoplasm of prokaryote, also in mitochondria and chloroplast of eukaryotes. This is 70S, made up of two subunits, 50S and 30S. Eukaryotic ribosomes, 80S, occur in cytoplasm of eukaryotic cells. Each ribosome consists of two subunits, larger and smaller subunits. 40S plus 60S together forms the 80S, and 30S plus 70S together constitute 70S. And here you can see the structure of a eukaryotic, that is 80S ribosomes. S is Rydberg's unit or sedimentation rate. Two subunits are held together by magnesium. Magnesium forms ionic bond with the phosphate group of rRNA of two subunits. And you know that the minimum concentration is 0.001 m. Concentration is required for the structural formation of ribosomes. Prokaryotic ribosomes have two subunits, a small unit 30S, larger subunit is 50S. 30S has 16S RNA and 21 types of protein. Larger subunit has 5 sRNA plus 23 sRNA and 34 types of protein. Total 2 types of RNA and 55 types of protein in the prokaryotic ribosome. Eukaryotic ribosome, 80S type. They are made up of 2 subunits. A small one is 40S and the large subunit is 60S. Subunit 40S, which is a smaller one, has 18 sRNA plus 30 types of protein. Larger subunit has 5 sRNA, 5.8 sRNA, and 28 sRNA, and 40 types of protein. So here the total comes to 2 types of RNAs and 70 types of proteins. At the time of protein synthesis, several ribosomes become attached to the mRNA with the help of a smaller subunit, and this structure is referred as polyribosomes or polysome or ergosome. The ribosomes move along the mRNA like beads on a string. Larger subunit contains peptidyl transferase enzyme, which is responsible for the peptide bond formation between the amino acid, and larger subunit has 2 sites, that is A site or acceptor site for tRNA, P site for the growing polypeptide chain.